The future of healthcare holds great opportunities for each of us. With major medical advances, we've access to more information and expertise about our health than ever before. Is it creating better patient care? Vantix wants to transform the delivery of healthcare by providing physicians and patients with the diagnostic information they need for better decisions, better patient care, better health. It all starts with diagnostics at the speed of now. The reference lab diagnostic model has fundamental flaws. Currently, a patient visits a doctor. 70% of the time, the doctor would order blood tests. Venous samples are drawn from the patient. The samples are shipped to a reference lab. The blood tests are performed in 1 to 3 days. Results are sent to the physician. Over the course of another 1 to 2 days, the physician reviews the patient file and the test results and decides on the necessary next steps. The patient is informed of the results over the next 3 to 5 days. Once the patient is informed, the prescribed therapy can begin. It's been at least 5 days, if not 10, since the initial patient visit. What Vantix Diagnostics is focusing on is how you do the same reference lab quality blood test in the physician's office in now time, connecting the patient visit with the diagnostic results, leading to the appropriate clinical decision at the time of the patient visit.
Today, we're all consumers of healthcare, and about 70% of the time when we go to a doctor, the doctor orders a blood test to confirm their diagnosis, sends you away to a reference lab, and 4 or 5 days later, this is obviously an inefficient process. And actually, about 40% of the time when the doctor orders the test, the patient doesn't even get the test done. When the patient gets the test done, about 60% of the time, the doctor doesn't even communicate the results back to the patient. Again, obviously this is ineffective and inefficient delivery of care. And what we've done using the Vantix biosensor is we've developed a way to do what the reference lab can do in the doctor's office in real time. The Vantix point-of-care system has 3 main components. The first is a disposable cartridge. The cartridge has the biosensor embedded in it, has the reagents already stored on it, and takes a fingerstick blood sample. That cartridge goes into a reader. The reader is a simple mechanical device that actuates the cartridge and generates the results with a simple user interface. And then the third and final component is the VLab. The VLab is a web-based application that connects the readers into the health information management systems on the back end, quality control systems, and then finally generates the results.
Well, the first product we're developing is the point-of-care system where we're measuring thyroid hormones. The 2 hormones we're measuring are free T4 and TSH. When we run these samples through clinical analyzers and compare them to our systems, we get the same results. We get the same equivalence in performance, we get the same levels of precision.
There are some core product attributes that you need to be able to hit in order to be truly disruptive in the point-of-care market. Initially, you have to meet the reference lab standard. That means you've got to be able to perform like the labs do today. Then you need to be able to get breadth of menu. That means being able to do general chemistries and immunoassays all on the same platform. It's got to be easy to use. That means that you don't need a trained operator to run it. It's got to be able to go from sample collection all the way through to claims processing and health information management systems integration. And then finally, you've got to be at a price point that you don't change the current reimbursement structure that's in the market today. We view our competition as the reference labs. And what we're trying to do is meet that reference lab standard of performance in the doctor's office in real time. There's other competitors out there which are other point-of-care technologies, but our goal is not to be incrementally better than them, but it's really to be disruptive and change the reference lab model.
It produces the results so quickly because of the measuring system we use. It's an electrical output signal. So we're able to combine that with simple biochemistry to produce a very rapid electrical output on the system.
In this building, we produce the sensors for use on our point-of-care systems and our research systems. The production of sensors has been investigated to make sure it's a robust process, so we're constantly producing the same sensors each time to the same standard with good quality. We've used a variety of techniques, especially Six Sigma techniques, which is a an industry accepted standard for developing production lines and manufacturing processes, so that we can assure our customers and other teams that they're getting the same product day after day. The screen printing process is similar to commercial processes used elsewhere. We use medical grade equipment downstairs to screen print the sensors. We have a large production capacity and it's very flexible. We produce a variety of sensor types using the same equipment. During the polymerization, we apply a conducting polymer onto the surface of the sensors, and this adds value by making them much more reproducible, much more sensitive, so that when we put the other reagents on top, they will react in a much more controlled way to the analyte being tested. In the BioDot process, we apply the biochemicals onto the surface of the sensor. These biochemicals will respond to the analyte that we're wanting to test for. The BioDot will very reproducibly apply a specific amount of reagent to each electrode on the sensor. These are then dried, treated before the sensors are cut up into individual units for use in the cartridges. We produce different formats of sensors. The process is flexible so that we can produce one format for handheld, another format for point of care. We've also developed experimental types for evaluation and for special applications.
The VR2 is an open access 12-channel device that is applicable to many assay formats. The device consists of 12 sensor combs for developing multiplexed assays, easy-to-use reader system with control software, USB connection, battery or USB powered. The portable device provides results in minutes versus hours for currently used reference lab ELISA assays.
We can change the biochemistry that's going onto the sensors very quickly. In a lot of cases, a lot of assays are antibody based, so it's a matter of applying a different antibody specific to the thing you want to test. So that means that we can use reagents that are currently out there, so say antibodies that are specific to tests, and apply that rapidly onto our system. So it's a matter of transferring those antibodies, for example, onto our system and optimizing the test.
Vantix is a privately held US company with offices outside Boston, MA and in Cambridge, UK. The US team comprises engineering, quality, regulatory, marketing and corporate. The UK team has expertise in sensor manufacturing, assay development and optimization, contract research support and biomarker development. Current priorities are distribution of the point-of-care platform in the European Union, India, Russia, China and Brazil. Co-development of markets such as retail clinics, biomarkers and veterinary. Sales and marketing of the VR2. The Vantix system is one core system with multiple commercial applications. The major market is the physician office, where the rapid provision of results can facilitate faster clinical decision-making, improved patient compliance and greater patient satisfaction, all of which can lead to improved clinical outcomes. Vantix Diagnostics, a well-established company poised for growth, has the capabilities to transform healthcare by delivering diagnostics at the speed of now.